Hey there folks, and welcome to the final video for Chapter 12, DNA Technology and Genomics. When we study genomics, now that we've done a bit on DNA technology, when we study genomics, we are looking at the entire genome for a species. All of the DNA it is possible, and all the different varieties of different alleles it is possible for that species to express. And the more we understand the genome of each different species that we are able to study, the more we understand not just about that species, but everything it is related to, and the more we understand about how DNA is structured, how it functions, and some of the similarities and differences between our own DNA and other species. In fact, studying our own DNA, the Human Genome Project, is still the most famous genomic study in the world. It was a long-term study. It took scientists from all over the world years to slowly sequence and understand all of the different varieties of DNA that we can see in human beings. But the information that we get out of this is invaluable. We discovered whole new things about DNA that we weren't familiar with before, and now we understand not just that they exist, but we also understand something better about what those types of DNA do for our DNA overall. Things like telomeres, which are sections of of repeated DNA, and transposable elements, which are whole sections of DNA that can be copied and pasted throughout the DNA chain without altering its function. The location and size of these types of repetitive DNA tell us something about the local DNA function, what those genes are for, where change is possible inside our DNA without altering its function, and where the DNA absolutely must be a specific way without mutation. And the studies go on. We still have certainly not understood the function and the sequence of all DNA for all species on this planet. These genomic studies will go on for a very long time. But again, every time we understand one more species, we understand just a little bit more about DNA at large. This diagram to the right, for example, is what it looks like to try and map out our human genome. That is one intense diagram, and a lot of work went into creating it, and there's a lot of information we can get out of it. But that's a whole other field of biology. We make a career out of studying that. We didn't just make a career out of studying genomics, however. This was such a massive undertaking. In order to really understand all the DNA of a species, we needed whole new DNA technology in order to handle this stuff. The things like the Human Genome Project went on early enough that we still only had fairly early tools for getting the job done. Early DNA sequencing techniques were logical and thorough, and they worked, but they were slow, and they involved various different steps of figuring out an individual's pedigree and their family variety of DNA, and then starting to sequence all of the DNA that they could get from those individuals. But because of the demand for better technology and more efficient techniques that new genomics projects were demanding, companies and scientists developed a whole new type of DNA technology just for DNA sequencing of these genes. This is called the whole genome shotgun method. Descriptive, right? This means that instead of taking one section at a time, you take the whole genome all at once, and you start sequencing right away and just sort of like a jigsaw puzzle, find out where the pieces fit. The old method worked, don't get me wrong. It was logical, it made sense, it definitely got the job done, but it was much slower. The whole genome shotgun sequencing method is much faster. However, it is also less precise. So there's definitely pro and con to using this method. Studying genomics has also given us better understanding of DNA and of ourselves in a couple other ways. For one thing, it's given rise to the field of proteomics. While genomics is the study of all of our genes and our DNA, and what section of the DNA encodes what gene, proteomics is the study of what whole full protein sets one genome encodes. That is to say, for all of the genes in this genome, what are all of the proteins that those genes are encoding? Between the fields of genomics and proteomics, we are learning more constantly about what's in our genes, what does each gene do, and not just what specifically does each gene do, but what does each gene code for. So eventually we'll have a much richer understanding if, say, someone or a specimen of some species has a really intense amount of particular protein that cells are making, we'll be able to find the genes in its DNA that direct the production of that protein and have a better, more detailed, real understanding of what's going on from the DNA in the nucleus all the way out to the entire organism. We've also acquired a better, richer understanding of our own evolutionary history. Humans are kind of in an interesting position because we are an animal that studies trying to understand itself and where we come from over evolutionary time. We're sort of like nature's philosopher, looking at ourselves, trying to understand where we come from, so maybe we can figure out where we're going. Part of our study of our own genome has given us an ability to understand a little bit more about where we have come from. By understanding the human genome, the modern day human genome, we've also been able to look back at DNA samples from our ancient ancestors, like the Neanderthals. And by comparing the Neanderthal genome to the modern day Homo sapiens human genome, we can see how our DNA has changed over time from that of our evolutionary ancestors. And perhaps eventually this will help us plot and track forward our own evolutionary pathway, and we can understand better how we have been changing over the course of our evolutionary lineage. Maybe someday this will even give us a better ability to even discuss, let alone predict, where our evolutionary changes will take us in the future. It's a fascinating thing to think about. Essentially, we are inside the experiment of evolution and trying to understand it from the outside at the same time. We're very lucky to be able to do this and to be able to build the tools that might let us do it a little bit more successfully. In the meantime, ladies and gentlemen, this is going to wrap us up for Chapter 12, and I'll see you next time. Bye, folks.